Hello, all you students out there that are absent or serving quarantine time. I wanted to go through what we did today so that you can stay up to date because some of this stuff is a little bit involved, I guess. It can be a little difficult, especially if you're missing out on things. So this was today's homepage. We were again continuing to look at the structure of DNA, but more so the things called transcription and translation. Hopefully you watched the last video that I had made that talked about some of those things. But so we're going to practice some transcription and translation, kind of review what I sent you guys for yesterday or what was linked on there. This little cow activity had a cow on the front of it. And then we started working on a transcription translation lab, which I think I sent that, or it was linked on yesterday's homepage. So anyways, let's talk about practicing transcription and translation real quick. Let me jump over here. Let's try this. Again, I had already discussed some of this stuff, but I'll just recap it for you quickly. Maybe. Taking a long time. Okay, finally here. All right, so we had talked about this idea of what's called the central dogma of biology, which is how life essentially works. Well, I guess let's work our way backwards. Living things are actually made of protein. This would be skin, hair, muscle, bone, tissue, tendons, right? Basically everything that makes the structure of a living thing is protein, and lots of chemicals inside. So that the information for making proteins, again, proteins are made out here in the cytoplasm of a cell by ribosomes. The ribosomes are reading a code of a molecule called RNA, and they're translating that into a protein, which is a chain of amino acids that we get. We get amino acids from what we eat and take into our bodies, and our cells use those to build our own proteins. And the RNA gets the information from the DNA that's inside the nucleus here. So again, the flow is, the code for making the proteins is our DNA. Messenger RNA, it's called, copies sections of that, takes that information out to the ribosome, where then the protein is actually assembled. The amino acids are put together. So, yeah. So the first step of going from DNA to RNA is called transcription. And then the second step of going from RNA to protein is called translation. So that's again, that's what we're talking about right now, is transcription and translation. So, all right. So let's go back and talk about how that works. What were we looking at? Oh, okay, well, let's just go back here. So today we did this transcription translation practice. Okay. So how this works then is, so again, the DNA is copied into something called messenger RNA. That's called transcription. And then the ribosomes will translate that messenger RNA sequence into amino acids. And that, again, is the protein. So let me enlarge this a little bit here. Where is the button? There it is. Okay. So I'm going to try to write on here. Should be fun. Okay, so the way it works again, so the messenger RNA is just, it copies by using complementary bases or nucleotides. So again, for messenger RNA, if this is a T on the DNA, it's going to have an A that pairs up with it. And there's an A here on the DNA. The messenger RNA is going to have a U, and then the C is going to have a G. So this is called a codon of messenger RNA that's complementary to TAC is AUG. And then you just continue doing that. G would be a C, C would be a G, G would be a C, this is a G, an A. This A is going to have a U, a C, 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 A, C, C. Okay, so then that's how you would do the transcription, making the messenger RNA. Then you would be looking these up on a chart. And again, if you go back to yesterday, I believe, and that's not going to let me do that. Oh. All right, well, I'll get rid of that. Okay, there we go. So, that's not what I wanted. it's like watching my grandma trying to run a VCR back in the day. I'll figure it out here eventually. Or I won't. And again, that's not what I want to do there. I hope you're enjoying watching me screw around here. How did I get that thing up there? Okay, well, maybe it's back to working again. I don't know. Okay, so here we go. So again, we transcribed that, and we have, again, yesterday. There we go. So yesterday I gave you this thing, this chart that you needed, and then, so let's open that up. That is where? Okay, so there's a square chart that you can use to look up these, what are called codons. So again, I go back to here. So the first codon is AUG. And so to look that up on here, you would find the first base on the left, the A, and it tells you that it's in this row somewhere. And then you look up the next one across the top, it's a U. So you come intersect the A and the U, it tells you it's one of these four. And then you go over to the far right side for the third base, which is G, come back to here, and that tells you that, okay, that AUG is methionine. So you're going to come back here and put MET right here, because the first amino acid coded by AUG is methionine, MET. Okay? So, and then the next one is CGC. So again, we'll look that up on the chart here. So it's CGC. So again, you go C here, it's in this row somewhere. G, it's in this column. So this row and this column, it's one of these four. So CGC, it's this one right here called arginine. So come back here, I'm going to put in ARG. Okay. And I want to show you how to use the circular chart because I actually prefer that one myself. If you scroll down here, Stupid thing. Okay, scroll down here and you have this chart. So let's go look at the next. Okay, the next codon is GGA. And then this one, so you start in the middle. So you go here, G, then here, G, and then A. So you go G, G, A. It's this thing called glycine. So I'm going to go back here, and I'm going to put G, L, Y right here. And then I'm going to go look up UCC. You can start in the middle, UCC. It's this one called serine. So I'm going to put S, E, R. So again, this is called translation. When you look up the messenger RNA codons, this is sort of kind of how the ribosome would do. It's like reading a recipe and then cooking up something. So you read the AUG, and that tells you you need to get methionine. You read CGC, you need to get the amino acid arginine. GGA, you get the amino acid glycine. UCC, you get the amino acid serine, and those together are starting to build a protein. Okay, let's do the last couple. CCC, C, 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 it's proline. So I'm going to put P, R, O here, proline, and then ACC, ACC, it's threonine. So put this down. It's THR. Okay, so that would be, again, how you do that. So I would go ahead, if I were you, and ignore this thing that says tRNA, but just go ahead and do the same thing we just did. Write out the messenger RNA. Again, it would be, again, here it's going to be complementary. A with T, A with T, G with C, and then take that AAG, look it up on the chart. And AAG is lysine, and then you'd write down here for the amino acids, just write lysine. Again, ignore this tRNA row for now and just do like we just did up there. So I would definitely practice that if I were you. Okay. And then next today we started, actually we went over this cow paper activity. So that was this one. I don't know if that's what I linked on there or not. Yes. So that was this paper. So all I wanted you guys to do was this box right here, which was exactly what we were just doing. You just take these, you write messenger RNA, you look up the amino acids. So I do want to show you the answers to that. So well, let's go back here. And I'll pull up the answers to that activity. I think it's this one. Of course, it's the other one. All right, so there you go. So those are the answers if you want to check, practice it and check and see if you're doing it right. All these ones that I didn't fill in down here are the same as the human. So you could just, these are the same thing down here, glycine, isoleucine, valine, etc. These are the two that are different from the human that I highlighted. So, okay, so that was, again, so today we went over that. And then lastly, we started this lab about transcription and translation. And yeah, so I will link that to the homepage for you, and you can check it out and do the best you can with it. It should be, as long as, well, I guess you kind of need to print, but do the best you can. I guess if you can't print, you can try to, I don't know, maybe figure out, draw the things or something. I don't know. So anyways, I'm going to wrap up here because I know you're probably bored watching this and me trying to click on things and nothing working. So anyways, I hope you're doing all right, and I'll hopefully see you soon.